talking about fertilization. In the last few lectures, I have been talking to you about pollination, wherein pollen are transferred onto the stigma of a flower so that the next step of fertilization can take place. Actually, pollination leads to fertilization. Once the pollen grains are transferred on the surface of the stigma, they germinate here, put forth their pollen tubes, and then pollen tubes carry the male gametes to the embryo sac where female gamete is there, and then they fuse together to form a zygote. Now, fertilization involves a sequence of events, and these events start right from pollination and then they go till the entry of pollen tubes into the ovules. And this sequence of events from pollination till the entry of pollen tubes into the ovules, that is, this is a step before actual fertilization takes place. This is what we refer to as the pollen-pistil interaction. The pistil on which the pollen grains they land, it screens the pollen grains, and only those pollen grains which are very vigorous and compatible, only those are selected for fertilization. Now, pollen landing on the stigma are diverse in origin. They can be belonging to the same species, which we refer to as the conspecific pollen, and of course, there are pollen which are from other species. These are called as the heterospecific pollen. Heterospecific pollen grains are in general prevented from taking part in fertilization as they are not recognized by the pistil, they are rejected. Pollen of all other species are inhibited at the stigma, and if they are not inhibited at the stigma, they are probably rejected at the style level. Now, this is a very simple diagram which is elucidating all the steps which are involved in pollen-pistil interaction. So, the first step is when the pollen lands, then it adheres to the stigma, then of course it imbibes water, it gets hydrated, it puts forth a small pollen tube, then pollen germination, that is what we refer to as pollen germination. The pollen tubes, they elongate and then they enter the styler canal, they reach the embryo sac, they reach the ovule wherein the embryo sac is present, and then of course they enter the embryo sac through one of the synergids, and then the fertilization takes place. Now, a very close interaction occurs between the pollen grains, or sometimes it is the pollen tubes, which are the gametophytic partners, and between the pistil. So, pollen grains are the gametophytic partners, the pistil is the sporophytic partner. So, there is an interaction between these two, and there are different peptides, there are different proteins which are secreted. Extracellular components present in and on the wall of the pollen, stigma and style, they are the ones which are involved in this recognition reaction. That is, pollen are recognized, the pistil recognize the pollen which it will allow to germinate. So, once the pollen grain is able to germinate, pollen tubes are guided towards the micropyle of the ovule by there are different types of attractants which are secreted by the synergids, and the pollen tube enters the embryo sac through one of the synergids, and once it enters, it discharges the two male gametes which are required for fertilization. Now, all this process of pollen-pistil interaction, this is basically to select the suitable type of pollen grains. So, there is a continuous interaction which is happening between the pollen grains and the stigma on which the pollen grains have landed. Conspecific pollen grains, they activate the physiological processes which allow the pollen to germinate and hence the pollen tube to grow. There are certain species which are self-incompatible, where the self pollen are inhibited on the stigma even after recognition. Otherwise, all conspecific pollen, the self pollen, they are able to germinate on the stigmas. Once the pollen grain is able to germinate, the interaction is now between the pollen tube and the transmitting tissue. Transmitting tissue is tissue which is present in the style. So, it is through this tissue the pollen tube is going to reach the ovule. The evolution of male gametophyte in the form of pollen and the retention of female gametophyte inside the ovule, this has made seed plants independent of water for fertilization. As you would have known that in lower plants, water is essential for fertilization. So, this is a very crucial step in the evolution of land plants. The pistil screens the pollen and permits only the right type of pollen to enter the ovules for fertilization. That is what we have just been talking about. That means if it is allowing only certain type of pollen to germinate, it is rejecting at the same time the heterospecific pollen grains which land on the pistil. So, the pistil is basically, it is the home of female partner and it has a series of locked doors. Now, any locked door will open with only a specific type of key, otherwise it will not open. So, we imagine that pistil is the home of female partner and it has a series of locked doors, and each of these locked doors which can be opened by only conspecific pollen which have the matching keys to those doors, otherwise it will not open. There may be many pollen which can be trying to open, but they will not be able to open. Now, this is one diagram which shows that the pollen which are conspecific, they are able to open the locks of the pistil because they are able to secrete certain peptides which bind with the components present on the stigma, and then those locked doors are opened and the pollen are able to germinate, whereas in the second one you see that they are not able to germinate. So, in the process of self-incompatibility, self pollen grains are definitely recognized by this pistil and they activate physiological processes, but these physiological processes, they inhibit the self pollen at the level of its germination, or if not at germination level, it could be later during the growth of the pollen tube. So, what happens here is that the self pollen are not able to effect fertilization. Self-incompatibility can also be said as the inability of a hermaphrodite plant to effect fertilization upon self-pollination. Both the sexes are present, the anthers are there, the pistil is there, so the pollen, they even land on the stigma, but they are either not able to germinate, or in case they germinate, the pollen tubes are inhibited in the styler canal and they are not able to effect fertilization upon self-pollination. So, this is what is called as self-incompatibility. Discouraging fertilization from self-pollen. So, if there is no fertilization happening with the self pollen, obviously there will be some other pollen which will help in the fertilization. So, discouraging fertilization from self pollen and encouraging fertilization by outcross pollen, it leads to a greater genetic diversity. Thus, when we talk about self-incompatibility, evolution of self-incompatibility, flowering plants have become 100% outbreeders by the involvement of pistil, leading to enhanced genetic diversity, because there is a cross pollination which is involved in this phenomenon. The number of pollen grains which are deposited on the stigma is generally many times more than the number of ovules which are available for fertilization. Because if you remember my lecture on pollination, we had discussed that most of the times, pollination is like it's a hit and miss phenomenon, though the species, they invest a lot in producing huge numbers of pollen grains, because the chances of the right type of pollen landing on the right stigma are very less. So, The number of pollen grains which land on the stigma are many more than the number of ovules which are available at that time, which can be fertilized. So, all compatible pollen which are landing on the stigma may not necessarily be able to germinate and find an ovule to fertilize. Sometimes they do germinate, but then what happens? The pollen tubes, they have to stop growing, or there is a competition between different pollen grains, and those pollen grains or with the pollen tubes which are more robust, they are able to reach the ovules faster than the other pollen grains. Hence, the pollen, they compete with each other to succeed in fertilization. More vigorous pollen, they reach the ovules earlier, and of course they affect fertilization. Less vigorous pollen, they are then eliminated in this competition or in this process. So, this process of elimination is often referred to as sexual selection, and it leads to better varieties which are produced. Then there is another phenomenon which is called as the pollen viability. Now, the ability of pollen to deliver functional male gametes into the embryo sac is called as pollen viability, right. So, huge amount of pollen grains is produced, number is very, very large, but all the pollen grains which are produced, and at whatever stage, they may not be able to deliver functional male gametes into the embryo sac, and the pollen generally show maximum viability soon after anthrodehiscence. In some, the viability can last for to about 30 minutes, whereas in others it can be for many days. So, viability is dependent on intrinsic nature of pollen species and is also dependent on the environmental conditions. Two-cell pollen generally retain viability for a longer period as compared to three-celled pollen. Okay, three-celled pollen, they lose their viability very fast. Two-cell pollen species like Leguminaceae, they can remain viable for several weeks. So, imagine that three-celled pollen, in case of Poaceae, the viability lasts for only 30 minutes. So, after that, if the pollen lands, it will not be able to effect fertilization, and in Leguminaceae, the viability can last up to several weeks. Another concept which you have to know at this stage is the receptivity of the stigma, called as the stigma receptivity. Now, like there is the ability of pollen to transfer male gametes for fertilization, there is also the ability of stigma to support the germination of viable and compatible pollen. Pollen viability and stigma receptivity are their respective peaks in freshly opened flowers, and that is the time which is most appropriate for fertilization to occur. The period for which the stigma retains receptivity, it varies greatly. It can again be from a few hours to a few weeks. Pollen wall and stigma surface components, they play important roles in pollen-pistil interaction, including pollen recognition followed by their promotion or inhibition. Now, as we have already discussed, that the stigma is going to recognize the right type of pollen which are landing on it to effect fertilization. So, this is recognition of conspecific pollen grains. Conspecific pollen grains are recognized by the pistil, and this activates different physiological processes which facilitate the germination of pollen and hence the pollen tube growth. Now, if you remember that pollen grains, they have a two-layered wall, that is the intine is the inner wall and exine is the outer wall. Intine as well as exine, they contain extracellular components, okay, and these extracellular components are made up of carbohydrates, lipids, proteins, glycoproteins, including several enzymes, carotenoids and phenolics. The intine components are synthesized by the cytoplasm of the pollen itself. So, they are gametophytic in origin. the exine proteins and the pollen coat substances, they are synthesized by the tapetal cells and they are incorporated in the exine only after the breakdown of the tapetum. Because they are originating from the tapetum, they are sporophytic in origin. Now, this picture generally gives you an idea about the life cycle of angiosperms. You see that in a flower, this is a hermaphrodite or a bisexual flower where there are both types of sex organs which are present. That is, from the anthers there are pollen grains which are liberated at the time of anther dehiscence. They land on the stigma, they germinate on the stigma, put forth the pollen tubes. These pollen tubes enter the ovule through one of the synergids. The pollen tube discharges its contents and the two male gametes, they fertilize. One fertilizes the egg cell, the other fertilizes the polar nuclei, and there is formation of embryo and endosperm. When embryo germinates, a new plant arises. Now, we have discussed about the pollen-pistil interaction, that is, once the pollen it lands on the stigma, it interacts with the pistil and it is the pistil which decides the fate of that pollen, whether it will be allowed to germinate or it will not be allowed to germinate, or even if it is allowed to germinate, whether it can put forth the pollen tubes and these pollen tubes can reach the ovules or not. Now, this process of pollen-pistil interaction once over, this leads to fertilization. Now, what is fertilization? You would have studied about fertilization, wherein we say that it is the fusion of the male and female gametes to initiate the development of a new organism. Okay. Now, here in case of plants, this process is known as syngamy, right. You can see here that the pollen tube is discharging two male gametes into the synergid, and adjacent to the synergid is the central cell and the egg cell. So, one of the male gametes moves to the egg cell to fertilize the egg cell, and this is what is syngamy. The other moves to the central cell and it fuses with the two polar nuclei, right. Now, this is a quick recap of what we have been talking about, that is the various steps which are involved from pollination to fertilization in angiosperms. I will be talking about each of these steps in detail with you shortly. Now, the first thing is, whenever the pollen it lands on the stigma, see sometimes the wind velocity is high, sometimes the stigma does not have that much surface area to catch all the pollen which are available. So, the first and the foremost process is of pollen adhesion, that is, the pollen grains, whichever pollen grains, whether conspecific or heterospecific, which land on the stigma, these pollen grains, they have to stick on to the stigma. They need to adhere to the stigma so that they do not fall off, and if they fall off, they get wasted, right. So, pollen adhesion is the first and the foremost process, and it is a very important process so that the pollen they remain adhered or sticking around to the stigma. Now, adhesion is dependent on the type of stigma. See, I will be talking to you about the two different types of stigmas. One is the dry stigma, the other is the wet stigma, right. Proteins and lipids which are present in the coat of the pollen, that is, they are present in the pollen coat, they are the ones which are very crucial for pollen adhesion. So, pollen adhesion is dependent on proteins and lipids present in the pollen coat. Once the pollen has put forth itself and it has adhered to the stigma, now it is not able to dislodge from the surface of the stigma. So what happens now is that you would remember that when the anther pollen are shed in a very desiccated state, and so when these pollen which are in highly desiccated stage, when they land on the stigma, they do not have much water content in them, and they are also in a very inactive physiological state. Stigma exudates. Stigma, okay, there are two types of stigmas. I will be talking to you in a few minutes. Wet stigmas, they have the presence of some exudates on their surface. Okay, so stigma exudates and the papillae which are present on the stigma, they help in the hydration of the pollen. Once the pollen gets hydrated, metabolic activities they resume. Hydrated pollen, obviously because they have imbibed in water and they have also imbibed other exudates, these hydrated pollen they swell, leading to the rupture of the intine and the apertures. That is how now the pollen tube is going to come out, right. So, after pollen hydration there is germination. You see here that there are several pollen which can be seen in this picture, and some of the pollen have long tubes coming out of them. So, this process of putting forth of these pollen tubes by rupturing of the pollen walls and the apertures is what is pollen germination. A pollen tube emerges out through one of the many apertures once the pollen grain is fully hydrated. So, if you would recall, when we were talking about the pollen, there are different shapes of pollen grains, and of course, the number of apertures which are present on pollen grains is also variable. In utricularia, for that matter, just as an example, there are about 20 to 22 apertures which are present, but the pollen tubes need not emerge from all the 22 apertures. In general, the pollen tube emerges from only one of the apertures present on the surface of the pollen grains. So, here you see that there are some pollen germinating. Once the pollen grain has germinated, so what has happened? Germination means putting forth the pollen tubes. Now, once these pollen tubes have been put forth, the pollen tubes now they have to enter through the stigma, move through the style and eventually reach the ovary. So, entry of pollen tube is dependent on the type of the stigma. It is very easily able to enter in wet stigmas, because here the cuticle is already ruptured. Why cuticle is ruptured? Because in wet stigmas there are exudates, and these exudates they reach the surface of the stigma by rupturing this cuticle. In dry type the cuticle is not ruptured, it has to be eroded, and this is done by the cutinase enzyme which is present in the pollen grains. So, cutinase in pollen grains, it hydrolyzes the cuticle and the pollen tube enters in the intercellular spaces. Okay. So, we will continue the rest of the journey in the next lecture. Thank you.